you can help fight COVID-19.
The pandemic felt so overwhelming. Staying at home wasn't enough.
The novel coronavirus has humbled every nation and every individual.
But it doesn't have to be like that.
Maybe if you could, gather all the computers in the world and put them to work.
And to be able to do things that you could not do by any other means.
With your help, we can.
The Folding at Home project does exactly that.
Folding at Home is using its distributed computing model to create the most powerful supercomputer on Earth.
The collective power of millions of volunteers to aid COVID-19 research efforts.
And a grassroots example.
Your rig is helping researchers find treatments and vaccines faster.
We're passionate about this effort and the impact it is having.
We need people like you.
Working together can make such a big difference.
You can learn more at foldingathome.org.
Welcome to the 20th anniversary celebration of Folding at Home. For the first segment of this, we're going to actually be discussing the past and the history, the legacy, and the future of Folding at Home. And I'm joined by a few great members of the consortium. The founder, Dr. Vijay Pande, Professor John Cadera at Memorial Sloan Kettering Cancer Center, and the current director and professor at Washington University in St. Louis, Dr. Greg Bowman. So if you guys just want to introduce yourselves.
Yeah, hi, I'm Greg Bowman, current director.
So up front, Greg, do you want to give some opening remarks?
Yeah, I'm really delighted to help kick off this celebration of 20 years of science with the Folding at Home community. And just to give a little bit of history, as the name of the project implies, the original impetus was really to understand this process of protein folding, in which these molecular machines that are responsible for everything from muscle contraction to vision can actually self-assemble or build themselves. And this is really astounding because we're used to having machines that can perform specific tasks, whether they're microwaves for cooking food or jets for transporting ourselves. But it's not so common for these machines to also be able to build themselves. And so the process of folding really captured the imagination of many scientists over the years. And one of the challenges has always been that there's no microscope where we can zoom in and watch this process with the resolution that we'd really like to understand how it works. And so it became very appealing to try to simulate this process on a computer. But there we faced another challenge, which is that simulating the folding of many proteins on a single computer could easily take hundreds to millions of years. And so this led people like our founder, Vijay, to start thinking about, well, how could we get many computers to work together on these otherwise intractable problems? And so through a combination of physics-based algorithms and then folding at home, the idea was to empower anyone with a computer and an internet connection to join a planetary scale distributed computing and pool their computational resources to tackle these really difficult problems. And so this really enabled a lot of advances in protein folding, including making quantitative predictions that were verified in subsequent experiments, which is a holy grail for theoretical sciences. And part of the idea with going after folding was always that if we could understand this complicated process of self-assembly, then we should be able to use the same sorts of methods to understand related problems like the mechanisms of diseases and to design drugs even. And indeed, the technologies that were originally developed to understand protein folding have continually opened up new areas of research. And now the Folding at Home is research team is actively engaged in everything from understanding neurodegenerative diseases like Alzheimer's disease to many forms of cancer, as well as infectious diseases like Ebola and the SFTS virus, where we've even gone so far as helping to repurpose an existing drug to treat the FSTS virus. And we're just about to help kick off clinical trials before the COVID-19 pandemic started. And so this put us in a really great position to quickly pivot in response to the pandemic, and start using folding at home to understand how the SARS-CoV-2 virus actually works and to identify therapeutic opportunities for shutting the virus down, and even to contribute directly to the development of new antivirals. And so we'll get into more of the details on this later on in the program, but this was all really enabled by the truly astounding compute resources that the Folding at Home community helped us to assemble. And so with your help, Folding at Home became the first machine to break the exascale barrier, providing more than 5 times the performance of the fastest supercomputer in the United States.
When we chatted, you talked a lot about the influences that you had that inspired Folding at Home. One of them being Napster, right?
Yeah, I think it was an interesting time because, so just to set the stage, this was basically 1999. And so I'd just gotten to Stanford and was thinking about, given the great opportunity in front of me about being a professor at a place like Stanford with great students and postdocs that I'd hopefully be able to attract, what could we do that would be really impactful, something that would really be a major push forward, not something just incremental. And looking around and spending a lot of time thinking, actually for me, it was like a lot of just walking around the campus trying to think of like something that really would be that profound. And those things obviously take some time to sort of try to find your way through. And it was a lot of stuff was in the air though, and especially the internet was finally starting to become pretty dominant. And it was actually specifically Napster that got my attention because it was clear that a lot of people could get together, could be networked together in interesting ways, and could fundamentally change how a business or an operation was done. And I think that was really the what I saw in Napster, both from a technological point of view, but also from the point of view of almost being a movement, which very much Folding at Home has always had to be as well.
Yeah. So then how did it evolve from this inspired by Napster, this idea of a distributed computing project? How did the initial details get hammered out? How did the first work server come about?
Yeah, so there was a small group of us that got together to write the initial code, and actually it was an intern that was working over the summer, and my lab was relatively small at that point. Or there were just a few grad students, but actually the key people with the code were largely me, Jared, the intern, and Adam Bieberg, who had written a toolkit that could be used for building these types of platforms called COSM. And so Jared and I took COSM and built out the first sort of version of this with some help from Adam. And, in the early days, it was interesting because it didn't take much to really overwhelm the hardware that we had. We had, I think the first work server was, had a Celeron processor at like 500 megahertz with a 10 gig hard drive. And maybe like 2 days into the project, the 10 gig hard drive was completely filled. And even though everything was much smaller then and the work units were much smaller, it didn't take long for us to realize that there was huge potential here.
That's awesome, to fill up a hard disk in that short amount of time. But was it helped then by some early collaborations and partnerships, right? I think you mentioned briefly that CNN had gotten some interest and that you even met with Google at some points.
Yeah, so I think this was something where this has always had to be part of the effort, is that we need to get the word out. And it's not like we have a multi-million-dollar marketing budget or something like this. This is an academic project run out of initially out for many years out of Stanford, my lab at Stanford, and so we had to engage donors in all the different ways we could. And having press and CNN was really useful for getting early interest. And that, one thing leads to another. And we actually then, in the early days of Google, were chatting with Sergey Brin, and he had the idea that we could collaborate, and he wanted to help out Folding at Home. So in the early days in Google, there's this thing called the Google Toolbar, which is a widget that you could add to a browser, and it would make browsing easier. And actually, Folding at Home was built into the Google Toolbar, so they could turn on Folding at Home for people to use. And that was another big leap forward. And it was those types of collaborations with Google, with Sony being shipped on the PS3 that allowed us to continue to grow faster and faster.
That's awesome that it got incorporated into the Google toolbar. But yeah, I mean, you mentioned the PS3. I mean, that happened not too long after, right? It was that a smooth transition then to upgrade into the PS3 and.
Yeah, so and it was an interesting time because we were already doing everything we could to be ambitious with computer hardware. If you start to try to run on hundreds of thousands to millions of computers throughout the internet, you start asking, what can you do to be even more efficient about it? And GPUs were very much on our mind, and we were lucky that Pat Hanrahan was a professor at Stanford and one of the sort of leaders in the space. Actually, Michael Schertz, who was a grad student at the time, realized that you could start and was thinking about programming these things. And one of the challenges, though, even at that time, was that programming was programming before CUDA, that you literally had to write in the shader language. You had to almost describe this as if it was some sort of graphics calculation. Mm-hmm. And so it was really sort of very sort of bleeding edge kind of stuff then, and especially putting out a GPU client before CUDA. And we had great help from Pat and actually Ian Buck, who was a grad student in Pat's lab and who later on went to NVIDIA and developed CUDA and is now leading the efforts there. It was with that in mind, we kept on looking around. So GPUs were one milestone. PS3 was a natural milestone from there because the cell processor there was very GPU-like. And we got a lot of help from Sony, especially Noam Riemann at Sony, who led the development team there. And they did a ton of work, which allowed us then to be on PS3s, which that much further gave us a ton of compute power. And in 2007, it actually led to us getting a first sort of world record. And this time it actually was sort of framed as a Guinness World Record for being the most powerful supercomputer in the world. At that time, petaflop, which is like 1,000th of the speed that was of the exaflop, which was recently broken.
I have to say that early 2007, late 2006 was the time that I started looking into joining Vijay's lab. And when I first came to the lab site and saw this giant pile of PS3s everywhere and enormous monitors, I could tell that this was going to be an exciting place. I remember this is when the Rock Band party started. Is that right? Is that every Friday afternoon?
The PS3s have different uses, but the compute part was huge.
Multifunction computational platforms.
Absolutely, yeah.
But it also helped with recognition, right? And the kind of being able to engage users not just with the PS3 and it being on the platform, but also recognizability. You could be at someone's house with a PlayStation and they would and you could tell them about Folding at Home that was running on it. I think Greg, actually you have said something.
That's right. I remember being at a dinner party at a friend's house, and we had a group of like a dozen of us, and I was explaining what my research was at the time, and people were intrigued and a little bit skeptical. They're like, "Oh, I just got this PS3 and I'm having a blast playing on it. Like, but why would I run Folding at Home?" So I was like, "Oh, well, let's take a look." So they powered it up, and one of the things that Sony had been felt strongly about was that I think I had 8 cores essentially, and so they dedicated one of them to rendering the simulation in real time with really beautiful graphics with this map of the globe behind it that showed where everyone who was running on a PlayStation was. And so we spent a good hour or so after that with people zooming around the map, seeing little islands where people were running Folding at Home and asking questions about this protein they were watching jiggle around on the screen. And I think a bunch of people then went home and started participating. So it was a really fun experience.
That map that ended up in the Sony visualizer was such an amazing thing because it really helped drive home what a global phenomena Folding at Home had already become by that time. You could see, lights in even very tiny cities out in remote areas. And that was quite striking to me at the time.
Yeah, very much so. And I think part of it too is that it wasn't just an app, but it came shipped on the PS3s. So it was just always there. And I think that was a great introduction. And especially too, a lot of people, a lot of gamers, they don't play the games on like a little phone. They play it on a big TV. And so people would tell me that, Folding at Home was like in their living room. It was in their lives.
Yeah, so and that's and that's kind of continued on even now with everyone. I mean, people still ask us about whether or not we're running on the PS3, and unfortunately there aren't many PS3s around anymore to run on. But yeah, we're always open to running on as many platforms as possible. But I mean, one great thing that's come out of this, and I think you already mentioned it, kind of and Greg did too, is that this was the beginning of actually being able to make the scientific advances and collect the kinds of data that enabled that. And I know there were some great advances in identifying some of the first protein folding events and identifying those trajectories. I know Greg, you've been involved in a lot of that and developing these kind of network-based models of protein folding.
You want to take that, Greg, or I can give a little background too.
Oh no, go ahead, Vijay.
Yeah, I mean, so here's the problem that, especially for people who don't live and read the sort of technical side of this, is that breaking up something that was originally, what we originally wanted was one computer that was 1,000,000 times faster. That would be nice. But instead you get 1,000,000 computers. And you might think that's equivalent, but really it isn't. Like if you think about like any task you'd have to do in real life, if you had someone to help you, maybe you could get it done twice as fast. If you had 10 people to help you, could you get it done 10 times faster? If you have 3,600 people, could you get an hour's job done in a second? Probably not. Just all the communication and organization gets really hard. That much harder when you multiply by hundreds of thousands to millions. And so what we were forced to do were to really rethink the problem. And it's in the complete rethinking that led to developing Markov state model approaches. And I think, ironically, in the end, the Markov state model approach, which actually John and Greg were involved with at the very early stages, are, I think, a much more rigorous solution to the original problem. The original problem was maybe just have a movie of a single trajectory of what happened. But the Markov state model approach now actually led to a level of statistical rigor that even if we had one computer that was 1,000,000 times faster, I would still want to do it with that approach. It still sort of blows my mind that we were forced to come up with this answer. And in the end, I think it's just a better answer overall.
Yeah. So, yeah. Okay, go ahead, guys.
Oh, I was just gonna say I agree, and it helped with a bunch of other related problems too. I mean, it's hard to just take millions of protein structures on a hard drive and say, look at it somehow and say, oh, I understand. And so this approach really helped with that as well, and helped overcome some of the challenges of alternative approaches where you had to simplify things too much, like just looking at them on a 2D projection. And the states and rates idea really provided a way to deal with the huge number of dimensions that our brains don't necessarily have an easy way to visualize.
Mm-hmm. Yeah, so and it's great that we it allowed everyone to take these large numbers of trajectories that everyone helped simulate and integrate them into a more distilled, useful view. And I mean, it really does sound like a lot of the one big component of this was, again, going back to the idea of these GPUs that were running so many of these trajectories. And so what was it like developing kind of these first pre-CUDA GPU-based engines to run Folding at Home on?
Yeah, I mean, luckily we didn't have to stay pre-CUDA for too long. I think and so once the GPU infrastructure, software infrastructure started getting established, I think that went a long way. And actually, we got a lot of help from NVIDIA, and Scott Legrand helped with that when he was there. One thing that's interesting to think about is what happens to all this code. Well, it was, I think, such an important advance that then we took the code and wanted to make it available not just for Folding at Home, but for everybody. And so this has led to OpenMM, GPU code that many people here have contributed to. And we've done this in general. We did this with our ability to do fast molecular dynamics on GPUs with OpenMM. We did this with the network models with MSMs, with the hope that what Folding Home was doing was it was forcing us to sort of live at this bleeding edge, but that as we develop these tools, we could give them to other people. And maybe they don't run on Folding at Home, but they run on a cluster. And the thing about Moore's law is that what used to be sort of world record breaking not that long ago, like that petaflop, now a petaflop's really almost nothing. You could easily get that on a GPU cluster kind of trivially. And so the things that Folding at Home does today, I think sort of will be much more typical 5 or 10 years from now. And then that has been the history over the last 2 decades.
What really struck me about the investment in GPUs is that you jumped on the bandwagon so early that it wasn't clear. Certainly the performance on GPUs was very poor in the initial days, but you could see the exponential scaling that it was going to overtake CPUs at some point once the algorithms were sorted out. I just wonder about those very early days, like what was the thinking, and did you have any interesting experiences with melting hardware as you started to push the GPUs to new limits?
Yeah, and we had a lot of really strong people programming these GPUs. Vishal Vanathan, did a lot in the early days. Imran Haq, Peter Eastman, many others, and these were really hardcore programmers that are really trying to push things. The very first implementation that Vishal pushed out was what was written in shader language. So it wasn't even, it was pre-CUDA, and it was 1/4 the speed of the CPU version. And it's funny because you might think that would be a huge disappointment, but just getting it to run at all with numerical precision and working on the GPU was a very, very hard task. And actually, it was my colleague Russ Altman who I was actually very excited for his reaction, because when I told him it was 1/4 the speed, he was excited because he could tell that it was 1/4 now, but that it wouldn't take long. And I think that was that was sort of generally the feeling in the air, that if we could get it to work, the future was there. And especially GPUs have doubled in speed far faster than CPUs, especially of late. And so it's been the only way really to make huge speed improvements.
I actually remember Vishal's defense, and I remember when he was talking about one of the early versions, he was saying how because he was writing in shader language, and the assumptions that the hardware made is that the code could only simulate proteins with 1,024 atoms in it. And so he was very happy that one of the early model systems the lab took an interest in happened to have 1,024 atoms. And, so it's really come a long way, and, the extreme flexibility that it has now.
Yeah. And it led to a lot of jokes about NANDs. He named the first GPU cluster NAND cluster. GPU code then and still now, and Yutong can speak to this, is very good at producing NANDs. And programming GPUs is largely the fight against the NANDs creeping in.
Yeah, I mean, John, you've taken over kind of OpenMM development now, and you're kind of spearheading it. And so NANDs are still a problem then, but I mean, you've put CUDA, you've allowed CUDA to be utilized fully on OpenMM. So what's that been like to partner with NVIDIA and push forward?
I should be clear, I've done probably the least amount of work in terms of getting OpenMM to where it is today. It's really all of the folks that Vijay mentioned, especially Peter Eastman, who's been the software scientist and benevolent dictator for life for OpenMM, that's poured his heart and soul into making it an open tool that lots of people use. It's really a fantastic thing because it's also has a great Python API where you can just build stuff on it very easily, or in C++. So it's powering a lot of science. Sometimes you don't even know that OpenMM is part of what the application you're using is, because it's built in as a library rather than as a first-class application. So OpenMM had made a lot of progress since we last put out an OpenMM-based core. So my task was just to update our current compute cores for Folding at Home that use OpenMM to some of the latest versions that had a lot more increased stability. And then, it's funny that you mentioned Adam Bieberg being instrumental in the early days. Well, 20 years later, he's now at NVIDIA as the container GPU compute czar, and was also super helpful in us getting the CUDA client or CUDA enabled OpenMM core out into the world. And the good news is that this seems to have squished a lot of the NAND problems we had previously. It's hard and very interesting when you're testing a code that's going to run on such a heterogeneous set of hardware, right? OpenMM is one of these few academic pieces of code that is run on like a million different machines, and a lot of very different varieties of hardware and very different environments. You mentioned also, Vijay, Imran Haq ran a very interesting experiment at one point. Maybe you could just elaborate on that. It came down to, he was able to observe the altitude at which different machines were because of the cosmic radiation causing memory errors.
Yeah, he did some really creative things, and he was looking at GPU failures and memory failures. And a lot of this was, if you run on so many different machines, we want to make sure that our calculations have integrity. And he did various tests, and it was just kind of amazing that once you get to that scale, even things like cosmic ray hitting a bit here and there from a gigabyte of RAM in a million computers, eventually even these things come to play.
Sadly, Folding at Home's career as a cosmic ray observatory was short-lived.
Exactly.
Well, even pushing forward from the short period where you were able to measure cosmic background radiation, kind of 2010 onwards is when it really started to pick up scientifically, right? It went from just looking at protein folding problems to further exploring the horizons of what could be done with this kind of distributed computing platform.
Yeah. Well, and you think about the name folding is has been our name for the project, but the act of folding is really about how proteins change their shape, and there's so much about protein function that has that in mind, and so conformational change is a key part of this. And what makes proteins so interesting and so complicated to understand experimentally are the sort of myriad nature of these changes, which are really hard to see experimentally and to quantify experimentally. And they're critical not just for sort of basic biology, but that's largely how a lot of drugs work, is that the drugs will either induce a conformational change or stabilize a conformational change. And between those two, really understanding this is going to be a major step towards a much more rational ability to design drugs. And so I think, and Greg and John have done a ton of work here. I think that's been sort of our realization probably about 10 years ago, is that compute power was finally enough that we could get to timescales to simulate and sizes simulate that we could go after much bigger systems. And some of the first things we did were things like kinases, which are small enough that we could simulate them, but really important and really key cancer targets. And kinases, I think, even still today, become really interesting things to study, even though the power of folding at home now, 10 years later, can allow us to study really huge things as well.
Yeah, this is actually, really related to a question that we just got in chat from fishy_FAH. How do you decide what proteins/diseases to study? I imagine grants play a big role, but what else?
Greg, you want to take that, or I can take that?
That's a great question. Let me just speak from my side. So I'm at the Memorial Sloan Kettering Cancer Center, where primarily we study different kinds of cancers. And cancer is not just one disease, but it's any way in which things can go wrong that lead to your cells dividing out of control. Normally, they sense their environment. They know they're only supposed to divide a certain number of times in order to not cause problems. But tumors that will experience uncontrolled growth just have those checkpoints ablated by a mutation from the environment, or sometimes it's genetic. It's nice that Vijay mentions kinases, because essentially all my lab studies up until recently with the COVID moonshot is kinases. There are 500 different kinds of proteins in your cells that act like logic gates, where a mutation in a tumor can cause, or that causes a tumor, can cause the logic to not work anymore. So suddenly it's doing things, it's always on, and it's causing cell proliferation that causes disease. So often we're looking for different kinds of diseases where we're able to take what we've learned from folding at home at scale, where we can run hundreds of different kinases or different kinase mutants we see in clinical populations, and understand how those alterations lead to disease, and how we can design new small molecules that might shut down those kinases specifically, so that unlike normal chemotherapeutics, which just kill every dividing cell in your body, hopefully faster than killing the tumor faster than it kills you, these very targeted therapies can go after very specific vulnerabilities. And Greg is looking at this from another perspective. Greg.
Yeah, so I mean, I would say just in general that part of this is motivated by practical considerations. Like, cancer has a lot of impact on a lot of lives, so it's a very appealing set of problems to go after. And part of it is by curiosity and things that surprise us, right? So, in one case, we was at a talk, and the speaker was presenting work where they were trying to target one part of a protein with a drug, and they found something that worked. And of course, that's the only place the drug could bind and work. And then it turned out, when they figured out experimentally how the drug worked, the protein had opened up and the drug had bound in a completely unexpected place. And so this really caught my attention as a completely unexpected conformational change. And as Vijay said, this could be a really interesting way to target proteins. So we've been exploring, how general these cryptic pockets, or unexpected pockets as we call them, are, and how we can target them. And this kind of theme of surprise shows up a bunch. So in another one, there's a protein called ApoE that's strongly implicated in Alzheimer's disease. And this protein has 299 amino acids, so 299 chemicals linked together form this protein. And it turns out that if you just change one of them, this can increase your risk of developing Alzheimer's disease by up to 15-fold. And so that's really surprising, and actually the strongest genetic risk factor for Alzheimer's disease. And it's just changing, tens of atoms out of many thousands of atoms in this protein. So again, this is another, kind of surprise that such a small change could have such a big impact, and kind of called to us to try to understand what's going on, and then to generalize this to apply to other systems where related things happen.
Awesome. Yeah, and it's also kind of interesting to think about. I mean, you mentioned that there's 299 amino acids in ApoE, but how, even relative to what Folding at Home was simulating maybe, 10 years ago or 15 years ago, I mean, that's already a huge jump in scale, right? In the size of the proteins that can be simulated, or...
Yeah, that's right. I mean, my first graduate work was on a protein with only 35 amino acids. So barely a protein. My wife is a developmental biologist. When I, showed it to her the first time, and we were dating, so it was kind of flirtatious, but she laughed at me and said, "That's not a protein." And now we're doing things with 3,600 residues, like the spike of the coronavirus, on thousands of times short, 1,000-fold longer time scales. So things have really come a long way.
Yeah, I think especially like for me is when we could do kinases and GPCRs, I think now we are covering a large swath of space, and both of them conformational change so critical, and so many drugs were hitting them. And that was maybe 5-10-ish years ago we started that, like 2012 for our first GPCR paper. So I think now it's very much about just going for more of it in every direction, longer speeds, bigger sizes, and just lots of different variations. And John, I don't know, maybe we'll talk about this later, but just even all the numerous calculations you can do for just all the different drug tests as well.
Yeah, I'll be talking a little bit more detail about how we're using Folding at Home to address the pandemic and the COVID moonshot by a few different technologies we can now run on Folding at Home to help the chemists which molecules to choose first and make those first to drive forward drug discovery projects.
Awesome. So, kind of as we talk about what we're doing now and where we're going in the future, and we can kind of shift focus briefly to that. This is a great question from Kelvin Simulator on our chat. Before COVID-19, what were the current challenges that were being addressed, and what will be the direction that you go towards after COVID as well?
Yeah, I think this is one of the beauties of Folding at Home, is that it's a really general platform that can be applied to so many problems that are all based on the same, fundamental physical, chemical, biological principles. So in my lab alone, before the pandemic, we were doing a bunch with, both everything from Ebola virus to, antibiotic resistance and Alzheimer's disease, and covering quite a broad range of applications, but again, with this unifying principle of protein dynamics and conformational change.
Our lab has remained very focused on kinases as a very rich set of drug targets. Vijay mentioned that between kinases and GPCRs, you've got maybe 80% of all active drug discovery projects right now. And there's a lot of need to develop new drugs in these areas because there's still only a handful of molecules in our arsenal for kinases, for example, for cancer. So right now, if you get certain kinds of cancers, there might be a good drug to treat it, which is fantastic. But for other kinds of cancers, there's just really nothing out there yet. So there's still a strong need there. The pandemic has really showed that, I guess necessity is the mother of invention. We could do things with folding at home we hadn't appreciated we could previously, including help prioritize small molecules to build on data sets that have been generated. So one of the exciting things is that all of the infrastructure we've built for supporting the COVID moonshot, for example, in free energy calculations can readily be applied to all of the programs that Diamond XChem, we'll hear from them a little bit later through Frank van Delft, is collecting. This is a fantastic experimental resource in the UK where they use $1 billion synchrotron to collect structural information about various disease targets for which there's no good molecules yet. And they use this fragment screening approach we'll hear in more detail to find starting points that you could then use to rapidly identify molecules to first understand the biology and then to make therapies against these particular diseases. They run several dozen projects a year that are public in order to help de-risk discovery projects and accelerate drug discovery in these areas. And we're excited to be able to continue to help them as we deploy the infrastructure in an automated manner to support their projects in the future.
Yeah, so I mean, even further than that, looking forward with folding at home and necessity being the mother of invention, as you said, John. What are some technologies that you folks are excited about developing? I mean, I know that there are some of us have been working in areas of deep learning and artificial intelligence, or a variety of different other technologies. So what are you folks excited to see?
One of the great things about OpenMM is that it's such a modular platform that it's now there's a whole new field of machine learning potentials emerging where the ease of working with machine learning frameworks like TensorFlow and PyTorch and JAX have made it possible for someone to very easily build new models that can do amazing things. And the models are now being built to predict the potential energy of a small molecule, perhaps in a different conformation or interacting with a protein in a manner that can very closely mimic quantum chemistry, which is usually much more expensive, like orders of magnitude more expensive. So there's these models like Annie, for example, which are still a little bit more costly than the molecular mechanics potentials we use, but can get you near quantum accuracy and can learn from data. We've recently shown, for example, that you can train them to both quantum chemical data and to experimental free energy data. So now the models are not just static, but can also learn as you go along and gather more data. And these tools are being integrated into OpenMM right now through plugins, so we'll be able to deploy them on Folding at Home. So this idea of being able to continue to use Folding at Home as a machine that can learn from its experience and integrating machine learning and adaptive calculations deeply at all levels is going to be very exciting because it suddenly allows us to do a lot more work with closed cycles of experiments to gather more data and then making more predictions. So I'm really excited about using Folding at Home adaptively in that sense.
Yeah, I think, these, going bigger in all these scales that Vijay mentioned is a super exciting opportunity. And, just to put things in perspective, the Folding at Home community has created the biggest, most powerful supercomputer in the world. And we've pointed it all at a virus that only has 28 proteins. Right, we're still not seeing everything that these proteins can do. And for comparison, the human body has something like 30,000 genes, and there's lots of variants from swapping around parts on the proteins that these are created from these genes. So there's still a lot to try to go after, especially when you start then thinking about all of the genetic variation between different people, for example. And so, one of the things I think is exciting is starting to, look at these different variations on proteins, and as efficiently as possible, and trying, so by brute force we can start, simulating a handful, on the order of a dozen different variants of a protein, for example, maybe 100. But then also continuing to make the algorithms that are running on top of folding at home more efficient. So we've been doing a lot with what are called adaptive sampling algorithms, where instead of just letting these simulations run on everyone's computers, which are basically doing this random walk that doesn't have any knowledge of what it's done before, and so it might be repetitive and do the same things over and over again before moving on to something new. We can actually get there and intervene and say, oh, we've already seen this, so let's stop that simulation and continue this simulation over here that's wandered into some new region of conformational space where we don't have as much data as we'd like. So this allows us to get another couple orders of magnitude speed up by just keeping the computers focused on doing what's most useful.
One of the exciting things, in addition to thinking about how we can get longer trajectories or map out conformation spaces, folding at home has traditionally been focused on a single protein or maybe a couple of interacting proteins. But now that cryo-EM, so cryo-electron microscopy, is changing the way we think about and see the microscopic world structurally, we can suddenly get giant complexes of many proteins interacting. And thinking about how we can scale folding at home to this scale is also really exciting. One of Vijay's former students, Brooke Cusick, has recently put out a really nice paper that shows how we can use machine learning to take data from folding at home simulations at the atomic scale and then scale up to very coarse-grained simulations that still recapitulate the same kinds of interactions at larger scale. So I'm also super excited to see how we can scale this up to start simulating whole viruses or large interacting subsystems or signaling systems. There's a lot that can be done.
Yeah, no, I'm super excited about all the potentials that we have and where we can go from here. Really briefly, I'm just aware of our time, but because I have the three of you here and we've got a couple of questions on this from Valentine Sidersky and Regtor. I apologize if those are not the correct pronunciations, but basically, one asked, what would be the best way for a PhD student in an experimental lab with little computational experience to start integrating folding at home into their science? And Regtor asks, how would you go about starting a PhD as a part of folding at home? So I mean, basically, advice for future interested young scientists.
Yeah, I think applying to the graduate programs where there's faculty who are involved in folding at home. So, Sloan Kettering, WashU, Temple University, KTH in Sweden, and Hong Kong University of Science and Technology if you're at the very early stages. And if you're already an experimentalist who's got a cool system with interesting conformational changes, we're always eager to pursue our curiosity and be surprised. So you should feel free to shoot an email and make a pitch, and we'd be happy to think about putting things on folding at home and collaborating.
John or Vijay, do you have any advice or?
I think there's great things that we can do with folding at home also to make it more accessible in the future. And we've already been talking about how we can, get more people access to folding at home through various different strategies. It's going to be exciting to see if we can build ways in which we can allow experimentalists to interact with folding at home in a way where their favorite protein might be easily modeled and give them some useful information. So if you have pitches about what you'd like to see or how we could help support your research in a more general way, without a direct collaboration, but maybe through better tools we could engineer, that would also be super fun to hear about.
Mm-hmm. And Vijay, this is kind of like a agglomeration of a few things I've seen in chat, but I think you might have some comments on this. Is what are you excited to see about potential further collaborations between these kinds of distributed computing networks and the startup space and the industry space?
Yeah, I think there's been a renaissance in compute in startups for drug design and other areas in biology. I think it's been a long time in coming. I think there's this famous book, The $1 Billion Molecule, which talks about the early history of Vertex, where there was a great deal of promise for computation revolutionizing the space. That was like 1980s, and just the amount of computer power then versus now is just so dramatically different. And so, we were basically getting faster, maybe 1,000-fold every decade. So, that's 1,000 to the fourth power. That's I'm trying to just even do all the zeros in my head. So it's just a remarkable difference in computer power. And so with that, I think now it's gotten to the point where people who are involved in this are in huge demand, whether we're talking about to do this in pharma, do this in startups, and actually people that were in my lab have gone off to do great things. Some of them have become professors like Greg and John. Some of them have gone off to found startups. Some of them have gone off to work in pharma. And I think in each of those areas, this intersection in compute and bio, especially at this type of scale and with this type of impact, has become of great interest. For startups, in general, it's like anything else that you want to be able to have an idea that is a really grand idea that's worthy of trying to build $1 billion or $10 billion company. And coming up with that idea itself is really difficult and takes some time, but then obviously that's where it just begins. And building a startup is probably one of the hardest things that anyone has to do. But that was probably where you'd start, is I would be really thoughtful about the idea and the team.
That's super cool. And I guess we have time for one last question, which is from Who Now 123. Does the COVID research and the simulation data that we're collecting now have direct usage, not just outside of, like, maybe not outside of the papers we publish directly, but is there a general purpose use for this, and how are we making it available?
Yeah, so I think there's a lot to be understood about how viruses work and how they, how do they interact with our immune systems to evade detection, and how do they get inside of cells that, is transferable knowledge between viruses, just going one level up from the SARS-CoV-2 virus. And so, we've been sharing all the data through platforms like OSF and the AWS public data sets project. So you can access anyone can access both our Markov models that try to, simplify things and keep, keep things small but informative in terms of file sizes, or even the raw simulation data. If people want to try out their own methods for making inferences from the raw data. And so we're hoping that we'll learn a lot both about the SARS-CoV-2 virus and directly contribute to efforts to combat it, as well as help just find general principles of how do these things undergo conformational changes, and what are the implications for other areas of biology like viruses in general.
Just a quick comment here. The data sets that Greg's lab has put on Folding at Home, you folks have generated 1 million times more data per protein than I ever saw during my PhD, which was studying protein dynamics. So if these data sets had existed then, it would be a totally different game. So we don't even know what we're going to learn from looking at just what we can learn about how proteins move just from these data sets. So this is really an unprecedented scale. I want to ask Vijay, like, did you think it would last 20 years? This is perhaps an unexpected success for your original concept.
Well, here's the thing, is that I think in the first few years, it felt like it could be over at any time. And in the first few years, there would be not uncommon that I'd be driving to Stanford at 11:30 p.m. to like reboot a server or something like that. And we were relying on people sticking with us, and people had a lot of choices. I think, there became in a sense, almost like Folding Home is like a baby, like our baby that, when you're first time parent, you're just afraid like to put the baby in the crib and this is going to survive. Who knows? But then once, Folding Home became more of a toddler to a kid, to a teenager, I think its survivals did start to feel like we could just keep this going indefinitely as long as we're doing good work and doing good parenting, so to speak. But it was the first few years that I think were really stressful. I would say like the first 5 years, and a lot of people in the lab did heroic things. It wasn't just me, and really went way beyond what you would normally do to get a chemistry PhD, to sort of just keep it rolling. And also, since then we were able to have the funds and the resources to professionalize things, and that went a long way. So yeah, I think the last 10 years haven't surprised me at all. I think once we got rolling, I think we got there. But yeah, the first 5 years, who knows? Like it could have fallen apart any time.
Awesome. So I am aware of our time, so I actually just want to take a second and thank Vijay, John, and Greg for joining us for this segment. This is super fun discussion, and I really enjoyed hearing all of your answers.
Thank you.
Thank you, Super.
Thanks. It's been a pleasure.